not going to be monitored throughout this webinar. So hi everyone, my name is Rima Cardle. I'm an environmental stewardship technician on the LakeSmart team for Lake of the Woods District Stewardship Association. LakeSmart is an environmental outreach and education program, which is unique to our area, and it is made up of a team of post-secondary students who have a focus in the environmental programs. Our goal is to share all the information we can to help people to live and play green at the lake. So we can see all too clearly that plastic pollution is a problem, but measuring specific harms of plastic in our environment requires specific assessment. Today, we have teamed up with IISD-Experimental Lakes Area to bring light to the growing issue and to show their new research project in hopes to discover how microplastics affect fish and other organisms in our freshwater systems. We have Dr. Mike Rennie from the Experimental Lakes Area who will be giving the presentation and helping to answer any questions that you may have at the end. Dr. Rennie is a fisheries ecologist and an associate professor at Lakehead University, where he holds a Canada Research Chair in Freshwater Ecology and Fisheries. He is also a research fellow with the IISD Experimental Lakes Area, where he conducts a large proportion of his research and oversees the fish population program. Dr. Rennie is interested in how disturbances changes ecosystem structure and how energy flows through ecosystems. So to start off today's webinar, we have a few polls to help us get a better understanding of your knowledge surrounding microplastics.
Just giving it a minute for everybody's votes to roll in here. Okay, I think I'll end it right now, and I'll share with you all. Looks like everybody, well, majority is around some. And then I have a couple more here. So yeah, this next one is how you heard about the webinar today. Awesome. So I'm excited to see that our social media is clearly reaching people. So that's super fun. And then the next one here, if you could tell us about yourself. And this one is multiple choice, so you can be more than one of these things. So lots of people have a special interest in the lake, which I'm happy to see. And yeah, that's super fun to see those results. And I think I have one last one for the beginning of our session here. And that is on your background on the experimental lake area research. Awesome. And looks like we have lots of folks that are familiar with it and some that aren't. So that's great to see. Thanks, everybody. And with that, I will pass it off to Mike.
Okay, great. Well, thank you for inviting me and getting an opportunity to speak to you all today. Unlike normal years where we'd have you all out at ELA, we're all hanging out in our kitchens and bedrooms and stuff and virtual offices to get together to talk today. So hopefully that allows you to kick back and relax and maybe enjoy a beverage of your choice while watching today's presentation. And so we're going to try and keep it a little bit interactive, thanks to the suggestions from these guys to work in some polls. So we'll make sure you're paying attention as we go along here. I'm going to focus most of the talk today on sort of, I guess, a bit of the path that led me to microplastics research and some of the work that we've done on Lake Winnipeg, which is really relevant for Lake of the Woods because it's right in the drainage. And then how that sort of led into the work that we're doing at ELA currently, which has really just started. So it's hard for me to give you too much in the way of results there yet, because like it's only been going for a couple of weeks. But hopefully the background that we give you will give you some sense of why we're doing that work in the first place, and a little bit about what we know so far, particular to the Lake Winnipeg drainage area. Okay, so we know that plastic pollution is a problem. There's a lot of it. Estimates that we have going back now several years peg it at about 270,000 tons floating around in the oceans, and which sounds shocking, but then if you think about that within the context of there's something like 5 to 13 million tons added annually by countries that are coastal to oceans, it becomes even more concerning. So even at these rates, like you can imagine that numbers increased by almost a third by this time. And so where does it go? All this stuff is, you know, I've kind of indicated that it's floating around in the ocean, but not all of it floats around. Seems like every time they go and do a survey somewhere, we find more. So here's some photos from submarine canyons several kilometers down in the ocean, and we can see there's, you know, Uncle Ben's converted rice hanging out at the bottom of the ocean. You know, we're looking for unique organisms that, you know, we've never discovered before, and yet we're discovering plastic waste, which is a little depressing. And there's some really good evidence, including some of the work that we've done on Lake Winnipeg, that seems to suggest that sediments are a major sink for microplastics. There's a lot, you know, when we try and look at how much plastic is coming into a system versus how much is actually there, there's a lot of there's a lot of that plastic that's unaccounted for, which indicates that a lot of it's ending up in the sediments. We also know that it ends up in wildlife. And so this is looking at sort of larger plastic particles, but here's a shearwater chick on Midway Island. And you can see, you know, the parents fly out, they find stuff floating on the surface to feed their chicks. Unfortunately, they're feeding their chicks a lot of plastics, and then they essentially starve to death because they can't get by on just plastic for a diet. This is a shearwater in New Zealand, again, looking at sort of stomach contents. This is an adult shearwater just full of plastic debris and particles, styrofoam and the sort. And it's in fish too. So this is a rainbow racer that was collected off of in the Pacific Ocean in the early 2010s. So we know that these things are ending up in wildlife, but there's still some knowledge gaps in terms of, like, you know, besides getting filled up on large quantities of plastic and starving to death, are there at low concentrations concerns in terms of how that might be affecting organisms? And I mean, not surprised, you know, for anyone who just woke up from a 1.5-year coma, we're in a global pandemic, right? And so, you know, I don't know when the last time you went to the grocery store was, but it seems like every time I go, when I look around the parking lot, you see evidence of improperly disposed PPE, face masks, gloves, I mean, you name it, right? And it's weird, I don't know about you guys, but I find that more often I find people wearing disposable masks than the reusables that we all seem to have started out with, that our aunt sewed for us. I'm not entirely sure why that is, but anyway, And that's led to some recent publications pointing out that these face masks that we're all using can be a major potential source for microplastic fibers in the environment. So if anything, you know, there's some concern that with the pandemic and the need for using personal protective equipment, which, you know, I'm not saying we shouldn't use it, but there's some concern that this is something that, you know, is going to contribute to the problem and accelerate the issue of plastic in the environment compared to what we used to have. Okay, so here's our first poll. We'll let people answer here. So the question is, what are some of the most common examples of plastic litter you see? So bags, like the grocery bag that's hanging in my neighbor's tree, personal protective equipment like the masks at the grocery store, food or drink containers, packaging materials, or other. So go ahead and vote when you get a chance.
Just going to give people a minute to answer this one.
Yeah, sounds great.
Okay, I'm going to wrap this one up.
Okay.
Yeah,
that's really cool. So, I mean, I think, you know, bags and food and drink containers and packaging materials, like that's all stuff that we would have seen a year and a half ago, which was still not all that great. But I'm struck by how many people are seeing PPE lying around now too, right? I mean, highly unscientific. I just complained about it, so maybe I influenced your answer. But at the same time, like there's a chunk that probably would have been less than 5% just a year and a half ago. So I think there's definitely some potential problems with that moving forward. Okay, onwards. So in my talk today, I'm going to, you know, the title of the talk was sort of about understanding the problem and finding solutions. And I've left a question mark on the finding solutions part because I'm hopefully going to leave you with enough information today to tell you that we're really still at the stage of understanding the problem, what the scope of the problem is, and not only that, but like what is the actual impact of this stuff in the environment. There's a whole aesthetic side of, you know, it's just, I'm not super excited about the idea of plastic particles in my drinking water or in my beer, or just hanging in my neighbor's tree. But there's a whole other level that we're trying to still get a good handle on in terms of what are the actual environmental impacts on organisms and impacts on whole ecosystems. And that's going to get us to the experiment that we're doing out at ELA today. So we're going to spend a bit of time talking about background exposure, how much plastic is just sort of out there in the environment since we've started looking. And we'll talk about some work that we've done in Lake Winnipeg and in the Red River, as well as at the Experimental Lakes Area. And that's really to sort of understand what the extent of the problem is, determining the fate of plastics in the environment. And, you know, so like once it's in the environment, where does it go? And that's really important to try and understand. And that's going to touch on some work that's been ongoing with a master's student at University of Manitoba. And then looking at what the impacts of gradients of exposure are on fish and ecosystems. And that's gonna really get to some of the work that we're doing at ELA to help us figure out what the impacts are on organisms and ecosystems. Okay, so most of the rest of this talk, I've talked about sort of plastic in general, but most of the rest of this talk is now going to be framed around the microplastics, right? And so you've all heard about microplastics. It's probably worth defining because I don't know that it often is. Basically, when we talk about microplastics, we're talking about small chunks of plastic, anything that's less than 5 millimeters in any dimension, okay? And oftentimes we characterize this stuff into different particle types. So you can have fragments, fibers, films, foams, like you can imagine packaging material or pellets. These pellets are often what you used to see in cosmetics and the whole banning of the bead was sort of really focused around that. So I think we're starting to see less of that in the environment, but these are sort of the major and common types that you'll encounter when you're doing this sort of work. And so we're gonna sort of now turn to Lake Winnipeg to try and understand. I mean, so back in, I guess, 2014, I saw a paper come out. It was early online, it was in 2013, where they had just done this sort of first survey of microplastics in the Great Lakes. And for anyone who's from Winnipeg or, or in Manitoba, when you hear about people talking about the Great Lakes and they, it seems somehow like Lake Winnipeg never gets part of that conversation. It's about the same size as Lake Erie. So at that point, I was kind of like, well, I feel like we should really try and do something about this. So I was lucky enough to get some funding from the Lake Winnipeg Foundation to start looking into thinking about how we might do a survey to look at what's in Lake Winnipeg and how that compares to the other Great Lakes. And Lake Winnipeg is quite unique compared to the other Great Lakes in Canada in that it's got a huge watershed. It's about 1 million square kilometers, and it's collecting water across 4 provinces and 2 states, 2 countries, right? And importantly for you guys, it's also collecting water that's coming from Lake of the Woods. There's about 7 million people spread out across this watershed. And so we went into this really asking, what is the extent of plastic pollution in Lake Winnipeg, and how does that compare to these Great Lakes where there's a smaller watershed, but the concentration of population and therefore potentially impacts is much greater. And even before we got started on this, we started getting sort of anecdotal information. So this Hetty Kling, who does a lot of the phytoplankton characterization in Lake Winnipeg, started seeing these blue fibers in some of her samples and was sort of like, hey, we're starting to see some stuff. And then Mike Stainton, who was also out doing sort of real-time characterization of water on Lake Winnipeg. This was up around the inflow from Grand Rapids into Lake Winnipeg in the North Basin, seeing these sorts of chunks that looks maybe like monofilament netting. So there was some idea that there was stuff in there, and it was maybe time to try and quantify what it is that's floating around in there. So we set out to establish sort of a baseline for plastic pollution in Lake Winnipeg because we really had no idea. And we purposely employed methods that would allow us to compare with other freshwater ecosystems that had used those same methods. And if you're interested in digging deeper, this is all published research, and I'd be more than happy to provide you with a copy if you tap me later. But it was largely driven by a summer student at ELA, Phil Anderson, who decided to, I mentioned I had these samples that needed counting, and he raised his hand. Sarah Warrack, who is now working with ELA and doing a master's on plastic pollution, also helped out with, I think, the lion's share of the samples from this work, and several other researchers, both at Lakehead and at University of Manitoba. So what did we do? We used what's called a manta trawl, and that's this thing in the water over here. And you can see it like Winnipeg, so you can see how green that sock is trailing behind it. We would tow it for about, we targeted half an hour. Sometimes we had to do less just because there was the net would fill up. For about 3 knots, you can't pull this thing super fast. Those samples were concentrated down in a cot end at the bottom of the net, and that was transferred to glass containers and preserved in ethanol for later analysis. These are the sampling sites that we selected on Lake Winnipeg. So we're using some of the traditional sampling sites that are hit or that are taken for water quality and other parameters by the research vessel in the Mayo. And so big thanks to Karen Scott for helping support all this work as well. So there are other attributes that we can link up to these different sites as well. And that sampling was done for 3 years. We did it from 2014 to 2016. When we got the samples back to the lab, they were concentrated down, and we got rid of all the biological material, all the algae and all the zooplankton, that sort of stuff, with a method called wet peroxide oxidation. So you take 30% hydrogen peroxide, which is nasty stuff, you heat it up to about 75 degrees, and that gets rid of a lot of the biological material. And what you're left with are the plastics, the non-organic bits and pieces in your sample. That was then manually identified. So we actually, this is what Phil and Sarah spent hours and hours doing, picking those samples out and getting what we thought were plastics into a different little jars. And then also we were able to validate that with electron microscopy, which at the time was sort of what people were doing. I think now you'd probably use either FTIR or Raman spec to characterize the plastics, but this at least helped us figure out, oh, that's a paint chip versus this is a organic material, or plastic, lots of carbon and hydrogen and that sort of stuff. So what did we see? So I told you we did 3 years of sampling, but we didn't see big differences across years. And this is sort of the general picture of what we found. So the size of the circle here gives you some idea of what the density is in the lake. So, top to bottom, they were averaging out around 200,000 particles per kilometer squared, which sounds like a big number. But if you think about that, boil that down to what it means in terms of a meter squared, and that's, in 1 meter squared, you're likely to encounter a particle of plastic, right? So, it's not a ton, but this is sort of the metric that we're using to compare to other systems. So here's our next question. How do you think the plastic that we found in Lake Winnipeg compares to the other Great Lakes? So lots more, about the same, less, or way less. If you think about it, you've got, think around the Great Lakes, you've got 35 million people living around the Great Lakes versus 7 million in the whole watershed for Lake Winnipeg.
Okay, I'll give folks another few seconds here to get their votes in.
Sounds good. Okay. Yeah, so lots of people had the same expectation that I did, which was that there'd be a lot less. You're in a much less populated watershed. There's not a whole lot of folks around, right? So you'd think there'd probably be less plastic around. So what did we see? Well, when you compare the densities in Lake Winnipeg to the other Great Lakes, and that's from this 2014 study that that got me interested in this in the first place, we find that the densities in Lake Winnipeg are on par with Lake Erie. There's 20 million people living in the Lake Erie watershed, and it's like 56,000 kilometers compared to 1 million, right? So that's much more dense. Although keep in mind what I said earlier too, that the size of the surface area of Lake Winnipeg is about the same as Lake Erie. So maybe that's a hint as to sort of where some of this plastic is coming from. Although if you look at the biggest lake in the bunch, here's Lake Superior, that's way down here, similar to Lake Huron, and big error bars around some of these lakes too. There weren't a ton of samples from that early survey on the Great Lakes. But if you scale up the densities of around 200,000 per kilometer squared to the surface of Lake Winnipeg, that works out to about 4.8 billion plastic particles floating around in the lake at any particular time. And that's those small particles that are less than 5 millimeters in size. So the other interesting part is we wanted to characterize exactly what it is that was floating around. And most of the particles of plastic that we were observing in this system were fibers. And so, yeah, like not fragments, not cosmetic beads or any of that stuff. It was mostly fibers. And this is maybe going to be important a bit later. So we followed that study up with trying to understand, well, what's coming in from watersheds, what's coming in from some of the major inflows into Lake Winnipeg. For her honors thesis, Sarah did a bunch of sampling on several rivers and mostly off the Red, which feeds Lake Winnipeg in 2016. And so these are the sites that she looked at, way down at Emerson, and then several sites through the city of Winnipeg at the Forks, where we have the Assiniboine River come in, and then right up into Selkirk. So what did we find there? So what was interesting is we found that there was some evidence that the densities of microplastics that we saw in the Red River increased as you sort of hit the city and went through the city. Interestingly, and we also, I should also mention, we took samples at the Nelson River at the outflow of Lake Winnipeg. And if you look at the densities going in, they can be quite high. They're variable, of course, but much lower than what we were observing at the Nelson River. So some evidence of an anthropogenic signal here going through the river. We also noticed a seasonal pattern in flux. And when I say flux, I'm talking about the densities in the water column that are adjusted for the cross-sectional area of flow. So we're talking about sort of particles per meters cubed per second. And I've also Z-scored these to standardize the values. If you don't know what that means, that's fine. We can talk about it later if you want. The important thing to note is that bigger numbers mean more plastics, smaller numbers mean less plastics. But we saw a fairly consistent pattern here, whether you're looking at any of the sites. The biggest pulses of plastic seem to happen in June, earlier in the year, and then that slowed down through July and October. So evidence of a potential pulse with the spring freshet. And interestingly, we also saw a correlation between flux and wastewater treatment plant discharge when we looked at discharge around 3 of the wastewater treatment plants in the city of Winnipeg. So when we have high discharge from the treatment plants, we also had high densities of plastics in the rivers, or high estimates of flux. Okay, so how much is the Red River actually contributing to what's in Lake Winnipeg? So if we can assume that the densities of microplastics in Lake Winnipeg are constant at around 4.8 billion, the estimated annual input from the Red River from those flux estimates works out to about 0.4 billion per year. Okay, so about 1/10 of what we're measuring in Lake Winnipeg at any given point in time. But what's interesting is our losses at the Nelson River are only 0.004 billion per year, right? So like 4 million per year, which leaves about 99% of the contributions that are coming in from the Red River unaccounted for. And so it begs the question, where's all that going? Our idea right now is that it's going to sedimentation, but we don't have, or I think there have been sediment samples taken, but no one's picked through them yet to see if we can figure out how much plastic's in there, to see if that explains the difference that we're seeing between what's going into the lake and what's leaving the lake. So between these 2 studies, we've been able to say that microplastic pollution in Lake Winnipeg is comparable to other Great Lakes, particularly Lake Erie, despite lower watershed population densities. So 7 versus 153 people per kilometer squared, basically. The pollution that we're seeing is primarily from fibers. We saw seasonal patterns of loading and evidence of retention in the lake, which we think is likely due to sedimentation. And another part of the work that Sarah did was also, she detected these particles in fish as well, some anywhere between 0 to 22 particles in the gastrointestinal tract on average. So this leads us into our ongoing work and where we're trying to go from here. And that's in terms of trying to understand what sort of the behavior of those particles are over longer periods of time, understanding what the amount of microplastic contamination is in remote regions, and also trying to understand what some of the sublethal or low concentration exposure responses are at the ecosystem level. And I'll touch on these as we go through here. So this is work that was done by Sarah Warrack, and it's ongoing at the University of Manitoba. She's defended, just needs to get those thesis revisions in. So well done, Sarah. Looking at the fate and behavior of microplastics. And I don't have a ton to show you here on this today, but we are seeing some interesting patterns in terms of the impacts of both nutrients as well as emergent plant densities on the settling and resuspension dynamics of plastics that we've looked at, and also some indication of how the resuspension and sedimentation of plastics behaves around the formation and loss of ice during the wintertime as well. So, and we're really looking forward to sort of getting that together in a publishable form. And that's all sort of led to where we are now with this, what we're calling the PELAstic Project. So, and this is really designed to understand the fate and effects of microplastics in aquatic ecosystems. And what we're trying to do by conducting this work at ELA is to do what we're really good at the ELA, which is to try and evaluate both direct and indirect effects on ecosystems. So oftentimes you'll find that one particular organism responds to a given contaminant, but it's oftentimes how all the other members of the ecosystem respond to, the loss of that one organism that really are telling in terms of what the overall impacts are on whole ecosystems. So I need to give a nod to all the people that are involved in this project. We've got lots. It's being led by Chelsea Rockman at the University of Toronto, myself and Diane Orihil at Queen's University. We also have an all-star cast supporting this work from various different places in the United States and Canada. Anyway, that's everybody. If I had more time, I'd walk you through them all. But it's been a wonderful group of people to work with, and for the work that we've done so far and where we're going, it's really exciting to see where we're going with this. And I'm remiss for not putting up the dozen grad students that are also involved in this, who are doing the lion's share of the work out there right now as we speak. So here's kind of our timeline. I'm going to talk a little bit about this reference sampling that we did back in 2019. Not surprisingly, 2020 was a bit of a wash. That was when we were going to do our mesocosm experiments that we're doing this year. So that's happening now. And this whole ecosystem experiment, this is where we're really trying to go here. This is all of the mesocosm work that we're doing this summer is meant to lead into this whole ecosystem experiment, which I think is now going to be delayed to 2023, but we're going to have some really good information coming out of the work that we're doing this year. So for those of you who don't know what ELA is, I'm going to maybe take just a quick pause to tell you, so you know. So the Experimental Lakes Area is a whole ecosystem aquatic research program that's was operated by the Government of Canada from 1968 to 2012, and it's located just between Kenora and Dryden. We're at the end of a 30-kilometer gravel road, so we're fairly well isolated from anthropogenic impacts. In May of 2012, the Government of Canada decided it was no longer willing to fund ELA, and thanks to an outpouring of support from the Canadian public, including lots of people around Lake of the Woods, and from the international scientific community, ELA was able to continue on. We now exist under the auspices of the International Institute for Sustainable Development as our own sort of not-for-profit organization, and have been working like that since April of 2014. And we're now supported by funding commitments from the province of Ontario, from the Government of Canada, and unlike our previous operating, unlike the way we used to operate, we're also now supported by private donors as well. Okay, so with that context, we're sort of in the middle of nowhere. One of the first things we wanted to do as part of this project was to try and understand what the background levels of microplastics would be in remote lakes. And so the information I'm going to present to you are from a couple of keen undergraduate students. One is Hayley McElraith from the University of Toronto, who's now going on to do a PhD in the UK, and Nadine El-Mariki, who did some of this work as part of our ELA field course that which we would be holding out there this August, if not for a global pandemic. So the last time we ran that field course, she did this as part of a project. So this is the work that we wanted to try and do, was look at the densities of microplastics in several different sort of categories of lakes around the ELA. We wanted to pick some lakes that had high activity, and we define high activity as being like, canoers go through there fairly frequently. So that was some of these larger lakes like Highwind, Hillock, and Tego, which are all on sort of major canoe routes. We wanted to look at medium activity lakes, which we defined as lakes that we've been doing research on for the past 30 years. So those were, I'm not going to go through the numbers, it's less important. And then we picked a series of lakes that were low activity. So these are lakes that we might have been to a handful of times over the past 5 decades, to try and understand whether or not there was any kind of impact of the amount of work that's being done on these lakes, either through research or through recreation, in the number of plastics that we see. The other thing that we wanted to do was to try and understand how much was coming in from the atmosphere, because as we looked in the literature, it became pretty evident that a lot of the plastics that are floating around are from atmospheric fallout. So we had 3 sampling stations for atmospheric deposition as well. So what did we find? So this is a summary of some of Hayley's data. So these are the low activity lakes, the medium activity lakes, and the high activity lakes. And there's really no difference across them. They seem to have very similar plastic densities. And this is what we saw regionally with the different locations for where we put out the atmospheric collectors. And those were not very different at all. The other thing I should point out is that of the particles that we were detecting in these samples, they were all, like the Lake Winnipeg samples, they were almost entirely dominated by fibers. And guess what the major type that you see is in atmospheric collections, in atmospheric fallout? It's fibers. And so we're currently working on developing this study a bit more and trying to do some mass balances to try and understand, based on what we're seeing in terms of atmospheric deposition, how much might that explain in terms of the concentrations that we observe in these lakes here. I suspect it's a fairly major fraction. I can't think of what else is going to be depositing plastics in some of these lakes that literally no one ever goes to. And yet we're still finding them there, right? So yeah, so there are, even in these very remote areas, we seem to have these very low levels of plastics. So what Nadine did is she actually went out with this manta trawl as well. So Hayley used a slightly different sampling method. This is using a manta trawl, which now we can use these densities to compare to all the other work that we did on the Great Lakes and on Lake Winnipeg. We did 3 transects in each lake. This is what the manta trawl looks like out of the water, and there's usually a cod end hanging on the bottom there. And so I'll ask you one more time, where do you think the ELA lakes fell in terms of concentrations or densities compared to Lake Winnipeg or the other Great Lakes?
Okay, I'll give folks another 10 seconds or so here.
That's good. Okay, so most people seem to be in the less or way less categories, a few in the more, and a couple in about the same. So let's take a look and see what we found. So it's about the same. So well, anyway, it was a little bit of a surprise to us. So here's Lake Winnipeg. So it's definitely less than Lake Winnipeg and Lake Erie, but I'm going to zoom in here. We've also got the other lake that's on here. This Havsgol is a lake in the middle of nowhere in Mongolia, which actually has people living around it. But surprisingly, the densities of microplastics that we observed in this one's our a reference lake where we've visited quite often. This lake here, 378, is where we're going to do our plastic. That's where we're doing our experiments now. But until we got there, we really hadn't done much work on that lake at all. The densities of microplastics in kilometers squared there in both of those systems actually is higher than what's estimated in either Lake Superior or Lake Huron, which is crazy. That in itself was very surprising to me as well. So here's where we are now. This is what we've spent all spring kind of building up to, and all of last year and the year before planning for, and busting our hump to get up this spring in order to get running. This is the first step in our experiment, which is a series of mesocosm experiments where we've added plastics to these different closed bottom mesocosms, which are 10 meters in diameter in Lake 378. We've done a gradient of plastic additions that go from 6 to 29,000 particles per liter. And we've got 2 control mesocosms in here as well to compare the results to. And if you look at in that front, the row on the left, I guess, the second one from the top, all that yellow there, that's all plastic. That's our high dose. And then our second high dose mesocosm is down here. That's in the bottom right on the back row, I guess, a second from the bottom. And this is what it looked like when we did the additions. You can see that after a while, all that's left on the top is the yellow. And that's largely because we picked these different types of plastics for differences in buoyancy. So the densities here, we're using, say, 29,000, but that's split equally between densities that are positively, neutrally, and negatively buoyant, okay? So after a couple of hours, what we found was that most of the plastic that was left at the top was the yellow. The pink was sort of in the water column, and the blue was at the bottom. And a lot of that pink now is actually settled out to the bottom as well. So there's all sorts of interesting stuff now that we're going to be able to do here, looking at fate of these particles as well, which is pretty exciting. I'm going to take a few minutes in the few that I have left just to talk about some of the work that my students are going to be doing related to this work. So one is Cody Veneruzzo at Lakehead University. He's a Master's of Science student. He's looking at how exposure to plastics are affecting the metabolic rates of fish. And so he's going to be using respirometry chambers and from the mesocosms to look at how they respond. He's also been able to take advantage of some lab work that we did earlier this year that's using the same concentrations that are or similar concentrations anyway as to what we're using in the mesocosms, where we looked at developmental impacts of these same concentrations on developing lake trout. So we had lake trout that were exposed as eggs and then looked at how they responded as they grew up into fry. This is work that's being done by Haley McLeod, who's a PhD student at Lakehead University, and she's going to go down to work with our collaborator at Ontario Tech University, Danina Simmons, who we're going to be looking at sort of proteomic responses. The cool thing is, though, is Cody was able to do some respirometry on these individuals, and we did see what looks like a bit of a dose-dependent response on the resting metabolic rate of these juvenile lake trout that we looked at the very end of the experiment. And where we saw the biggest difference, maybe not surprisingly, is at this highest dose. So that stood out from the rest. And what was also really cool is we had a second exposure that had the same high densities of plastics, but all of the additives were removed from the plastics, so that what we're adding to these mesocosms are plastics like you'd find in the environment with additives. And these additives are used to control the breakdown of these plastics from UV light, to add color. So it's very rare that you're going to encounter plastics in the environment that don't have things added to them to help them persist longer. So even though we're at the same high concentration that yielded a response here, we saw no response, which is really cool because this suggests that any response that we might actually be seeing in these fish anyway might be due to the additives that are in the plastics rather than the plastics, just the mechanical effect of having the plastics around. And so the next step is the ecosystem responses, and that's going to be done through monitoring these mesocosms this year, and then as we go forward into 2022 and 2023, preparing and actually conducting a whole ecosystem exposure to microplastics to see how the ecosystem responds. In conclusion, microplastics are everywhere. And so, and if you don't believe it, just from the work that I showed you today about Lake Winnipeg and the Experimental Lakes Area, people have climbed the Rocky Mountains and found some puddle at the top of a mountain, and they find them there. They find them in the Arctic Circle, and they find them in Antarctica. Everywhere we look, we find microplastics. So now the question is, how big of a problem is that? There's that aesthetic piece, which I think lots of people would agree, like we don't like just having plastic everywhere. It's kind of gross, it's unbecoming. But the bigger question of what's the environmental impact is still not really well understood. And so the research that we're conducting at ELA is really designed to understand that. And our group as a whole, the plastics group, is really tackling this through, not just through the whole lake experimentation, but using mesocosms to inform that whole lake experiment, as well as more detailed lab experiments that'll help us understand what's really going on in that whole lake. And I think there's really good potential for the results that we're going to generate from this work, as they often have from work that we've done at the ELA to guide environmental policy on microplastics. So what about the solutions piece? That was in my title, and I unfortunately don't have a whole lot to tell you about solutions, because in some ways we really need this basic research to help guide the environmental regulations around the release of plastics and microplastics in the environment. But does that mean we can't do anything? I don't think so. I think again, even just from that aesthetic perspective of like, I don't want to see plastic bags blowing around in the backyard all the time. I think there's things that we can do today that can help, even just from that aesthetic standpoint, to try and reduce our footprint with what's going out in the environment. And also too, I think another thing to start asking about is, can we develop technologies or best practices to avoid concentrations that are going to have negative impacts on ecosystems? And I think we're going to be able to figure out, based on the work that we're doing this summer and next summer, what those critical concentrations are going to be. So I guess here's the last question as to what you can do today. And so we can maybe take a poll and see what people think they might want to do here. So some examples of what you can do are some shoreline or spring cleanup efforts. I know I did a spring up to clean up here in Thunder Bay with my 9-year-old daughter, and I would say 98% of what that bag was filled with was plastic. Making more informed consumer practices, so avoiding plastic packaging or only recyclable plastics where you can. Properly disposing of PPE, or using or reusing face masks, and also if you can, help out by donating. We continue to do this work because of our amazing partners and our donors. And if you believe in a future where fresh water is for everyone, and if you're in a position to give, please feel free to join us today. And the link is there on the slide.
And I'll give our plug for tomorrow. Lake Smart is doing our first virtual shoreline cleanup. So our team's going to be out doing a shoreline cleanup. And if you would like to go out on your own and then either send us a photo or tag us on social media of you doing your own little cleanup, you'll get entered to win a bundle of red pine seedlings.
Oh, sweet.
So, yeah. Join us tomorrow if you can.
Decorations for the garden. Yeah.
Mm-hmm. Okay, I'll end the polling and share you guys what everybody said.
That's awesome. That's great. I think it'll be even if only some of us do what we said we might do in this poll, I think it's an important step forward. So that's great. Well, thanks everyone for listening today. I need to acknowledge all the folks and organizations that helped make this happen: the Lake Winnipeg Foundation, the Lake Winnipeg Research Consortium, and the RV Namao, NSERC, Environment Canada, MyTax, and Fisheries and Oceans Canada for funding, and so many people that I couldn't possibly list them all here. And I feel really, really lucky to be part of a group with this plastics experiment. It's probably, I would say it's been so far one of the best collaborative experiences I've had, and really looking forward to learning more as we move forward. And I'm happy to take any questions that people will have.
Well, I'll start with our first question here,
Okay.
which is a really good question. She's wondering if it's possible that the estimators of plastic in Lake Huron and Superior are vastly underestimated. So you're seeing, large amounts of plastic in our lakes that are super remote. Is that only comparable because the sampling is not the same in these other lakes?
So is it underestimated? Yes. And I would say it's underestimated across the board, because all of those, so the numbers that I showed you where we were comparing Lake Winnipeg to the Great Lakes and our ELA lakes compared to Lake Winnipeg and the Great Lakes, those were all sampled using a manta trawl. And that manta trawl has a 300 micron mesh. So we're collecting plastics that are between 5 millimeters and 300 microns. And if you look at the data, so the data that Haley McIlwraith had from our background ELA lakes, that was taken with a peristaltic pump. And that gets us down all the way to like 20 microns. And if you look at where the highest densities of microplastics are, they're actually at around about 100 microns. So I would say any of the data that's from the manta trawls probably underestimates exactly how much is there. However, we purposely picked that sampling method so that we would be able to compare apples to apples what the densities were in Lake Winnipeg compared to what we had in the Great Lakes and reported at the time. I think what it means, though, is that we actually need to get out there and do some sampling that'll help us characterize those smaller sized particles and understand, get a better handle on not just comparatively what's high, what's low, but using sampling that's going to capture the low end of those particle sizes. Does that change the picture in terms of what we think is more or less contaminated?
Right. The next one here is, what are the types of plastic added to the enclosures at ELA? Were they fibers or fragments or any of the other ones you mentioned?
Yeah, so we used fragments largely because we had to make or have produced this stuff to add into the lakes. So we essentially bought plastics that were like cell phone cases, essentially. And then those got ground up and milled by a company in the US, and then they sent us all the milled plastic. So we're using fragments. And I think if you look at impacted systems like Lake Ontario or areas where you have high densities of anthropogenic impacts, fragments are a large proportion of what you see there. The overwhelming densities of fibers is what we tend to see in sort of these remote areas and where you don't have a lot of impact. So we picked fragments largely out of convenience, but also it is still reflective of what you might see in more heavily impacted systems. And then we also took the additional step of using 3 different types, so that differed based on their buoyancy in the water column.
Right. And it was different buoyancies of fragments, though, not different types.
That's right. That's right. And so, and based on the size characterizations, I think the peak densities of what we added is around 100 microns, which is very similar to what you see in the environment. So,
cool. We have one more here. Since most of the microplastics are fibrous, don't most of these come from clothing? Which I've heard as well that your laundry machine is a huge contributor to microplastics, especially in around lakes.
Well, it certainly can. And if you look at sort of what's coming out of wastewater treatment plants, that's largely what you see there. Like what comes out in the effluent that isn't retained in biosolids is often fibers. But then the larger question is like, well, how does it get into the air, right? So arguably, maybe some stuff makes it through the lint trap in your dryer and that shows up. But I think there's I don't know, if you added up potential inputs from all the dryers in the world, I have a feeling that you'd probably still find there's more floating around in the atmosphere than we could account for from that. I think it's a contributing factor. I don't think it's the only factor. And much like sort of some of these complicated and sort of forever contaminants that we keep hearing about, like PFAS and stuff like that, they can, if they get up into the upper atmosphere, they'll distribute globally. And I think that's what we're looking at with plastics right now. There's a lot of work that needs to be done in terms of trying to understand what sources are and what potential non-point sources are for this into the atmosphere. But I think the evidence is starting to point towards that's where a lot of the stuff is coming in these, especially in these remote regions. So.
Do you have an idea of a source of an atmospheric pollution like that?
I don't know. It's a good question. Manufacturing processes. Dryers is for sure one. I mean, when you think about it, like every time we sort of look at a little more closely at what's contributing to the problems, like there's some really interesting studies showing that, like, even just like the wear from tires, right? When you have a big pulse of rainwater in cities, you get this big pulse of microplastics that can be traced back to tire wear, right? Yeah, it's a good question, and I'm not the person to ask that, unfortunately. So.
Okay, thank you for trying. We've got one more question here. It's in Winnipeg, was all the data collected from the top layer of the water, or were there samples also taken from deeper in the river? And then there's a second part here. Is it possible that the numbers are significantly higher with larger plastic particles as opposed to microplastics in the river?
It's a good question. Part A, yes, it was all surface sampling. So again, with the Mantitrol, you're only getting larger particles, but you're also only sampling from the surface. So I think that when we started developing sampling with that, the idea was like, well, yeah, of course all this stuff floats, right? But obviously not all of it does. And I think even the floaty stuff will sink eventually. I think some of Sarah's work has showed that pretty well as well. Once you get biofilm development and that sort of stuff. So if we had accounted for larger particle plastic, would we get a different answer? That's a good question. I don't know. I mean, you could probably, I don't know. I think that there might be some interesting ways to look at that. If you had some kind of, like, you could leave sort of a collector out for a very long period of time with a very large mesh size, storm grates, stuff like that, you could probably see what you could get from sort of the macro plastic piece there. That's a good question. I don't know. There's been less, I would say, where you see a lot more data and information on large particle plastics is with shoreline surveys and that sort of thing, particularly around the Great Lakes. I haven't seen a lot in terms of Lake Winnipeg or even along the inflowing rivers. I have seen less in terms of sort of macro surveys in lakes and rivers generally. So yeah, okay. Compared to microplastics anyway.
Right. And then we have got one last question here. And it's, are you changing the types of plastics that you add to the lake? Oh, wait, sorry. Are you changing the types of plastic added to the lake for the whole lake?
Experiment, probably. Yeah. So right. So right now, we're using these three types of sort of positively, neutrally, and negatively bland, right? And we've done a gradient of concentrations there. So that's sort of our goal this year, is to try and figure that out. We're hoping to run a series of mesocosm experiments next year. So once we find sort of what is that concentration that seems to sort of like generate responses in several of the different ecosystem components, we could say, okay, well, our concentration for the whole lake experiment should probably be around here. we want to do some additional experiments at that concentration with mesocosms next year to try and say, okay, well, which of the particle types is generating the response? Is it entirely due to, you know, the floaty stuff, or is it entirely due to the stuff that sank? And try and fine tune that question before we do the whole lake experiment. So it's a good question, and I think the answer is we don't know yet. And our plan is to be able to conduct a series of experiments next year in order to help us answer that question better. As to whether we use all three types that we're currently using, or whether we want to change things up and potentially just limit it to the one that we think is causing the most problems.
Okay, and so of the three that you have, this is my question, sorry. Between the three types of plastic, do they all have different like composition? And do you have an idea of which one is going to be the most harmful in the long run, or?
I don't. They all have fairly standard additives in terms of like either UV stabilizers or even just color, right? Like to add color, those are often chemical additives that will leach out over time. And most of which are known to have some kind of like toxic effect in aquatic organisms. So yeah, and I think, and some of Cody's preliminary results are really intriguing there in terms of, you know, because I think one of the things that we've had a hard time separating is like, if you're a fish and there's lots of plastic around and you just pack your gut with plastic and you starve to death, that's pretty bad, right? And maybe that happens at really high concentrations, which is sort of a problem with a lot of the lab experiments that have been done to date. But at a low concentration, even if you're not consuming those plastics, if they're leaching toxic chemicals that are going to impact you, that can have impacts at much lower concentrations than just packing your gut full of plastic particles. So it's a good question. And I think it'll be interesting to see how this plays out over the next few years. We're really hoping to do both lab-based experiments that look specifically at the additives, as well as some additional mesocosm work, like I talked about earlier, that will hopefully help us figure that out a bit better.
Cool. And sorry, we just have one last question if we have time.
Yep.
It's, do the additives in the plastics you mentioned during the metabolic experiment enter the environment over time? If so, does it happen quickly or over many years? So I guess it's kind of along the same.
Yeah, that's okay. So we use the same particle types that we're using in the whole lake experiment here. Okay. And same additives. And so the exposure time, I mean, it's a good question. I don't know whether it's a pulsed release at the beginning or whether it's over time. We did take water samples and those got sent to U of T. So we will be able to do some characterization of the water in the tanks over time to see what the concentrations look like. So we'll have that information when it's time to publish. Yeah, I think that's all I can say. Well, and we are definitely collecting water in the mesocosm experiments to try and answer that. Like, how, do you get a big release and then it slows down over time? Does it ramp up over time? I'm not sure we entirely know what the dynamics of them are, and it probably is additive dependent. I mean, this is part of the problem with doing plastics research, right? You're like, I'm going to study plastics. Well, what kind of plastics? What size? What additives? Like, all of a sudden it spins out of control. So our approach here has really been to try and pick particles that we can work with, but that are still informative from sort of a regulatory perspective, so that we can say, look, these are the kinds of things that you're seeing in the environment commonly. Can we start to use this to inform generally how we want to treat this very complex contaminant on the landscape and in our waterways?
Awesome, thank you. And I'll pass it off to Cassidy here.
Sure, I appreciate everyone hanging on for the little bit of extra time here, but those were awesome questions and I enjoyed hearing the answers. So yeah, I just wanted to say thank you to Mike and thank you to all of our attendees for participating, and the recording of this session will be posted on our YouTube and social media later. So feel free to reach out if you want to follow up with anything. And again, thank you so much, Mike. That was awesome.
Well, thanks for everyone for attending today, and yeah, have a great afternoon, and Weekend in one day. Woohoo.
Thank you, Mike.
All right.
Thank you.
Take care,
I'm
everybody.
looking forward to catching up with you in a couple of years and seeing where it goes.
Yeah. Well, I'm interested to see where it goes too. That's great. Okay.
Have a
Bye,
good night,
everybody.
everyone.